Hello, good morning, everyone, and thank you for joining me for lecture today. We're going to be going over chapter 12, and the topic of this is meiosis and alternation of generations. And we have one overarching lecture topic that is left in the class, that is genetics and biotechnology. And chapter 12 gives you some background information that you need to know to be able to proceed with the next chapter. Some people may be more familiar with this or have more of a recent background because it's commonly covered in the high school biology classes. Okay, so we'll talk about two different types of reproduction, sexual and asexual, the two phases of meiosis and how they also correspond to mitosis and are similar to mitosis. And then finally, we'll go over alternation of generations. Okay, so first of all, asexual reproduction is going to be more like mitosis because you're going to get identical individuals as a result of the asexual propagation. Okay, so this would be like propagation from cuttings that we did in the early part of the class. For sexual reproduction, this is going to be more like meiosis because you're going to get typically dissimilar offspring. Here, you're going to need materials from two parents or perhaps the pollen and ovule from the same plant. Okay, but it's going to be as a result of fertilization. So for sexual reproduction, there are gametes that are produced in meiosis, and then the zygote is the result of the sperm and the egg, or in this case, the pollen and the ovule uniting. So if you recall in mitosis, the cells are going to get larger, and then they're going to divide as well. So there's an increase in the number of cells, there's elongation of cells, and then proliferation of cells by cell division. So for mitosis, you're going to get uniform individuals as a result. And you start out with a diploid individual, that is to say, an individual that has two copies of the genetic information. And then at the end of the mitosis process, you're going to have two daughter cells that are identical. They also perfectly resemble the mother cell unless there's some sort of error during the process. And they're also going to both be diploid. Okay. So they'll be identical to the mother. So asexual propagation could have been an adaptation for plants to be able to survive and carry on. Okay, there are examples of natural vegetative propagation. The one that we like to use is with the strawberry because it's familiar to many of us. And you can have strawberries that reproduce by seed, but they can also reproduce asexually, and they throw out runners, and the runners themselves can root and then throw out additional runners. And each one of those rooted plantlets can then become its own plant, right, and produce seeds or further asexually reproduce as well. Okay, so in ferns, spores are produced by mitosis. So this is a little bit confusing because we're talking about meiosis, and we'll talk about sporophytes. But in this instance, the author is referring to ferns, and those ferns would be produced by mitosis. Okay, and also if you take a cutting, a vegetative cutting, and then root it, that is going to typically give identical individuals, completely uniform. This could be an advantage. The reason why this is an advantage is because you're going to have more uniform growth, you would have uniform harvest time, and with agriculture, since we are trying to control the process, it may work better. Okay, but there could also be some disadvantages. Okay, let's take a look at sexual reproduction. So this is pretty common. There are some plants that do not do well from seed or that don't produce seed, but in general, most plants are going to have sexual reproduction. Okay, so in our flowering plants or our angiosperms, it leads to formation of seeds. And for the cone-bearing plants or the gymnosperms, then it also will lead to production of seeds within the cones. Okay, there are gametes that are produced. The gametes are the male and female generative cells, okay, the pollen and the ovule. And when those unite and fertilization occurs, the result is called a zygote. Oh, look, and it's our strawberry plant. Okay, so we can see here very simply that the asexual reproduction is essentially the result of mitosis as the cells divide and become more numerous. And then of course they can specialize, okay? And then it's asexual vegetative reproduction, okay? And in terms of meiosis, we actually have alternation of generations, and it's a more complex process, although it does also involve mitosis. Meiosis is going to be that driver of diversity. So the advantage of sexual reproduction is that it leads to more diversity, okay? It is possible to have purebred lines or inbred lines in plant breeding, but if we think about the advantages of diversity in the field, it is the case that if we have uniform, either purebred or asexually reproduced plants all throughout the field, if one of them is susceptible to a disease, then that means all of the individuals out in the field are susceptible to disease. However, if we have some variation, then that could lead to more of the individuals being viable. It gives them at least a fighting chance. Okay, so there's a couple of words that I want you to be aware of, and I don't want you to fall into the pitfall of messing these up here. Okay, so an allele is a specific form of a gene. Okay, and a gene is a piece of DNA that controls a certain trait. When we say trait, we could also use the word phenotype. It's going to be a measurable attribute that you can see. Okay, so this should be somewhat familiar to you from chapter 3. Okay, in diploid organisms, each cell will have 2 sets of chromosomes, one from mom and one from dad. Okay, so each pair of chromosomes, they're homologous. That means that they're basically the same. They'll be the same in length, the amount of DNA and genes carried, and also the location of the centromere. It's going to be near the center, but in some cases you'll have arms that are long and short. Okay, now the result of meiosis as opposed to mitosis is going to be 4 cells, and that's going to be the result of 2 divisions. So that's why we get 4 cells, but they have less genetic information in each cell. They're going to be a complete set of genes, but you're only going to have 1 copy. So you guys will see how that works. Okay, and then because there is some shuffling of the deck during meiosis, in meiosis 1, then you're not typically going to wind up with identical gametes. So that can lead to new combinations of genes. Okay, so the main question here that we can answer is, why don't the seeds look identical to their parents? Okay, so this may not be the case if you have just a pure monoculture and you have purebred lines. Okay, but think about an heirloom set of seeds. A really good example of this would be tomato heirlooms. So if you look at the tomatoes, they will be slightly different in size and shape, and you might see different colors, but overall they're siblings and they're very similar to one another. Okay, so a connected concept here would be a land race, which is a small population of closely related individuals. Okay, so you're going to see that there's some variation. So why is that? Okay, so let's talk about meiosis, because meiosis is essentially the answer. Okay, so prior to meiosis, then you're going to have a doubling of the chromosomes. And here you're going to have two identical DNA molecules that are held together by a centromere. It's going to be approximately in the center. Okay. And then in meiosis, there are two divisions. The first division is meiosis 1, and the number of chromosomes is reduced to half. But then in division 2, there's no reduction in chromosome number. So let's take a look, and you may see that this also reminds you of mitosis as well. So in prophase 1, we have the condensed coiled chromosomes, where they were first difficult to see under the microscope. Now they've gotten a little bit shorter and fatter. Okay, and the homologous pairs, which are a little bit difficult to distinguish here. We have one that kind of looks like an X, or a typical homologous pair, is held together with a centromere. Okay, then the spindle fibers, which are made of protein, are going to connect to that centromere, and then the nuclear envelope will dissolve and dissociate from the chromosomes themselves. Okay, and in prophase 1, crossing over will occur. Let's take a look and see what that looks like. Okay, so it literally is going to mean that parts of each of the homologous chromosomes, or sections, little fragments here, are going to form these chiasmata, which are kind of like bridges, and they're going to connect to each other, and they're going to swap pieces of DNA. Okay, so on the right here, we can see the chromosomes are lined up in pairs, and then they have these little parts that are crossed over, just like we see here in the schematic diagram. Okay, and you can further see this once you examine what's going on in metaphase 1, because here we have the sections at the end of these two homologous pairs here that have swapped. And it's very obvious because they're color coded, just to make it more clear for us. Okay, and we can see the same thing here on the telomeres of these two chromosomes as well, which form a homologous pair. All right, so in metaphase, you can see that these chromosomes are more nicely aligned. Okay, although the chromatids are not as easy to spot, and they just kind of look like little loops or little worms that are curled up here, we can see that they are nicely aligned at the equator in our micrograph as well. All right, and now we have full sets of spindle fibers that are ready to move and help these homologous pairs divide. This is the reduction division. Okay, here in anaphase 1, we can see that one of the chromosomes from each pair is going to go to a separate pole. So now they're being pulled apart. And in telophase, which is the end phase here, we have now the result, which is 2 cells and 2 separate nuclei. So there has been a division. And remember here in anaphase, these separate parts of the pair were pulled apart before this new cell wall was created. Okay, great. So for division 2, it's going to look a lot like mitosis. Okay, in prophase 2, we're going to see this familiar process and result where we have our condensed coiled chromosomes. They're more easy to spot under the microscope. In metaphase 2, you can see that the spindle fibers have grabbed a hold of those centromeres, and the chromosomes are nicely aligned along the equator here, okay, instead of being scattered all around. And then in anaphase 2, the centromeres and chromatids of each chromosome are separated. They're going to be pulled apart and migrated to opposite poles. Okay, very similar to what we saw in the first round. Okay, now in telophase 2, the chromosomes will become longer, they're thinner, they're harder to spot under the microscope. And then each group of chromosomes is going to get its own nuclear envelope to protect it. All right, and finally, new cell walls form. Okay, so this would be like a spore, which would be in the sporophyte generation at the end of meiosis 2. Okay, and you can see how this closely resembles mitosis, although there are some differences. The key difference here being in metaphase 1 or prophase 1 is where the crossing over occurs. And then in metaphase 1, you can actually see that fragments of the chromosomes have swapped between the homologous pairs. And here we have the alignment along the equator, which is the same as in mitosis. Okay, and clearly mitosis has less steps. Here in anaphase, in both processes, the pairs are being pulled to opposite poles. Okay, you can see here in anaphase 1 that we don't have the homologous pairs in the same way that we did in meiosis. And then the result of mitosis is 2 identical daughter cells. Okay, and they have, they're diploid and they have 1 set of the genetic information from mom and 1 set from dad, 2 pairs of chromosomes. Two copies of the chromosomes. All right, so here in telophase 1, okay, you can see that you have 2 cells, okay, but they contain still these homologous pairs. And there's going to be a further division than in metaphase 1. So now we're seeing the chromosomes getting lined up again by the spindle fibers along the equator. In anaphase 2, they're being pulled apart. And then in telophase 2, the result of meiosis is going to be 4 cells, and they have a reduced number of chromosomes. This is only going to be haploid, since these are gametes. They only have 1 set of the genetic information. Okay. And then, as you can see, because crossing over occurred, now we have new combinations of genes. You can see that there's a little bit of variation as represented by the color coding because pieces of the chromosomes swapped. And the pieces of the chromosomes, you could think of those as being the information that was handed down from the grandparents in a way. So now you're essentially swapping the maternal and paternal grandparents, little parts of them, okay? And then they're going to be carried on to the generation, which winds up essentially being the grandchildren of that plant. So if you've ever seen maybe in puppy breeding, sometimes people will say that the children will resemble the grandparents. And you also might hear that with people as well. Okay, and this could potentially be an explanation for that. Okay, and here is a closer look. All right, so now getting into alternation of generations. So this is going to be between the haploid generation and the diploid generation, or the gametophyte generation and the sporophyte generation. So for the haploid, we'll typically say 1 end. Then we're only going to have a single set of chromosomes. That is going to be a gamete. That would be like a pollen cell or an ovule. Okay, and for the diploid, on the other hand, you would have 2 sets of chromosomes. Now, this is just like any of the somatic cells, as opposed to the generative cells. And the point at which the organism becomes diploid is going to be at fertilization when the zygote is formed. Okay, so it should make sense that if you have your pollen and ovule, and both of those are 1N or haploid, and those two join together and form a zygote. Now you have two sets of chromosomes, or two copies of the genetic information, one from mom and one from dad. Okay, there are other options here though. So this is very simplified. We like to look at the diploid organisms. For one thing, it appeals to us. We can understand it. We're diploid, but also working with diploid plants like pea plants is just easier. It's a simpler model. But in nature, we also see polyploids. They would have many copies of the genetic information. Okay, a good example would be seedless fruit, which are triploid or 3N. You saw that the homologous pairs have to align for meiosis to occur properly. And because you have a triplet instead of a multiple of two, the chromosomes do not align properly and there are no viable seeds that form. Okay, and another possibility here would be bread wheat or Triticum aestivum, which is hexaploid. It has 6 copies of the genetic information. It's another good example of a polyploid. We also have octoploid strawberries, which I thought was very fascinating. Okay, so here's a diagram showing you what this cycle of alternation of generations looks like. Okay, so it will involve sexual reproduction. Okay, and it's going to go between the diploid sporophyte, which is the green on top, and then the haploid gametophyte, which is the bottom half here. It looks sort of beige. Okay, the sporophyte, as you can see, is going to arise from the zygote, and then it will produce a sporocyte. This is essentially a plant that is ready for reproduction, right? It's going to produce the gametes. So that's going to be at the inception of meiosis. Okay, and then the result of meiosis is going to be that it produces 4 haploid spores. So that's going to be just like we saw a couple of slides ago, where we had our 4 varying cells and they were gametes, right? They only had 1 copy of the genetic information, or gametophytes. So that spore will develop into the gametophyte, and then meiosis or mitosis will occur, giving rise to the gametes. Once those gametes are fertilized, then we have the zygote, and it's going to start all over again. Okay, so just a little bit more detail here. The gametophytes are going to develop from the spores, okay? And then the gametes are going to form from cell division or mitosis in the gametophyte. Okay. Then fertilization, recall, is the fusion of the gametes, or when the gametes are going to join together. And then from there, we get the zygote. All right, so we can see this in the protista, the fungi, and the plants, but it's really obvious in the plant kingdom, and it's something that we can readily observe. Okay, so in the gametophyte generation here down on the bottom, the first cell is the spore, okay? And then the final cells are going to be the gametes. Okay, any one of these cells in the gametophytes is going to have half as many chromosomes as the sporophyte. Because remember, the sporophyte generation is diploid and the gametophyte generation is haploid. It only has 1 copy. Okay, the first cell of the sporophyte generation, on the other hand, up here on top is the zygote. And then the last cell that's considered part of sporophyte generation is a sporocyte. All right. The change from sporophyte to gametophyte is going to be when meiosis occurs. So you can see that here. This is the dividing line between the 2 processes. And after the sporocyte, meiosis occurs. And from then on, those cells are gametophytes. Okay. On the other hand, the flip side of the coin here, we have fertilization. Once fertilization occurs, we no longer have haploid cells because those haploid gametes are fused together and they form a zygote, which is going to be diploid. Okay, and then you can see it just a little bit bigger for clarity. Okay, so a key point here about sexual reproduction, and one of its advantages is that it creates new gene combinations. Okay, the recombination or the swapping of the pieces of chromosome that happen in meiosis 1 prophase 1 is going to take pieces of the maternal chromosomes with the paternal chromosomes. So that makes sense. Now for the segregation, when the chromosomes are pulled apart and then packaged into 4 daughter cells, then that essentially is going to randomly assort the chromosomes, right? And then when the union of the sperm and egg occurs, we have unique combinations, okay? Because you could have, if you think of this in terms of outcrossing, you have different individuals that have unique combinations of genes from their maternal chromosomes and paternal chromosomes that they inherited, and so does the other individual. So now when there's a fusion of gametes, we have this random assortment and can see new unique combinations. The thing is, though, that we're not going to see something that wasn't there before. We can see unique combinations, but we're not going to get new genes out of the process of sexual reproduction. That would be the result of a mutation, which is a different story. Okay, so remember asexual reproduction, that's more like cloning. That's like what we did for tissue culture or propagation by cuttings. And we're going to get identical individuals, okay? On the other hand, sexual reproduction involves crossing over and independent assortment in meiosis 1 and meiosis 2. So we get new allelic combinations. Makes it more interesting, okay? But some crops are going to be more inclined to produce and be productive with asexual reproduction and others with sexual reproduction. But if we have the choice, sexual reproduction is going to have some advantages. All right. And then we talked all about meiosis. We talked about how meiosis and mitosis are similar. That could come up on the exam. But really, for the purpose of our background information leading into genetics, we're really just most interested in the crossing over in meiosis 1, although the random independent assortment is important as well. Okay, guys, well, thanks for joining me for this short lecture, and see you soon.